So our second speaker, who I believe is here, hopefully, is Morgan Beebe.
Yep.
Good. So a little quick introduction. So Morgan got his PhD from UCLA in 2007 under Todd Yates, working on X-ray crystallography and comparative genomics. He then had 4 years at Caltech in Grant Jensen's lab and developed his interest in an electron cryotomography, which is what he's going to talk about today. In 2013, he took up a post at Imperial College in London, and he's now a senior lecturer there. So I can see your screen, so I guess we're good to go. Okay, thanks, Morgan.
Great, thanks a lot, Martin. Can you also hear me?
I can hear you fine.
Excellent. Okay, guys, I hope everyone's doing well in these conditions. So I'm going to be giving a talk really on applications of cryotomography on my favorite system, which is the bacterial flagella motor. I don't anticipate many of you will be that sort of deeply interested in the details, so I'm going to try and skim over as much as I can the nitty-gritty of the biology and really try to put across paint a picture of how we are using tomography and what sorts of advantages it brings us. So at heart, what I'm really interested in is protein evolution, and the bacterial flagella motor, which you can see in action in this video, is an amazing model system for asking questions about molecular evolution. This is a cartoon of what the bacterial flagella motor looks like. So the flagella motor itself is a rotary motor with which is driven by proton flux from the proton motive force from the periplasm. The motor proteins, which are proton channels, are called the stator complexes. These are embedded in the inner membrane and reach up to grab hold of the peptidoglycan layer that's sort of a part of the rigid superstructure of the bacterial cell. Proton flux through the stator complexes, in some way, we don't know yet the exact mechanism, in some way drive rotation of the rotor component of the motor, which is called the C-ring. Now, the C-ring is linked to all of the axial components, and so when the stator complexes rotate the C-ring in turn rotates all of the axial components, including the extracellular flagella filament. The flagella filament coils to become a helical propeller, so the bacterium propels itself through the liquid media. Another core component, which I'm going to talk about a bit later, is the type 3 secretion system. So that's a technical phrase. Essentially what the type 3 secretion system is doing is it is pulling in, unfolding and exporting protein components. And those protein components travel through the hollow central axial system to assemble at the growing distal tip. So the type 3 secretion system can unfold proteins that then travel multiple microns through the growing distal structure. Now, needless to say, our favorite technique is electron microscopy, in particular, the sub-flavor of electron cryotomography. So what we're doing is we are flash freezing whole bacterial cells. This is the cell pole of a Campylobacter cell. And we then perform subtomogram averaging on the flagella motor. This is a flagella motor. Zoom in. It's noisy, so we play the trick of imaging many of them, superimposing and averaging them to get a subtomogram average. Right? So this is a fairly low resolution structure. The joke I made at CCPEM a few years ago was that we are talking about the highest resolutions probably in the entire symposium. We're talking about double-digit resolutions here, think the resolution of this subtomogram average is about 80 angstroms. Joke. So what we'll then do is we can then start using that as the basis for asking biological questions. So in this case, long ago, actually still during my postdoc in Grant's lab, we had determined the structure of the Campylobacter flagellomotor, and we started wondering what pieces were. And so we wondered in particular what this protein was. We hypothesized it was a protein called FLI, which is a core component of the type 3 secretion system. And so to test that hypothesis, we teamed up with collaborators to delete FLI, re-imaged via subtomogram averaging. Because we're not having to purify fragile assembly intermediates or disrupted structures, the bacterium still assembled the whole flagellomotor, just lacking FLI, and what we saw was this. The only significant difference was the absence of that blob. So pretty conclusively confirming our speculation that that density is FLI. Okay, so we're going to tell you two stories about what cryotomography enables in terms of our research. So first of all, cryotomography enables insights into flagellar evolution. This is the backbone of much of our research. So this is a phylogenetic tree of flagellomotors, and over the years we've been putting the pieces together and putting in context what the flagellomotors and different branches of this tree look like. So I'm going to very quickly run through this. E. coli and Salmonella are the model systems. These are fairly well characterized flagellomotors, and that's what they look like. So these are slices through subtomogram averages. If we start going further afield though, and imaging lots of diversity from different parts of the tree, you start seeing this incredible variety across the tree. They've all got a common core that looks like the E. coli or the Salmonella flagellomotor, but many of them have bolted on extra bits, extra proteins. And so this really fascinates us and really asks the question, well, what's the selective benefit? Because if we could answer what the selective benefit is, that would then help us dig deeper into, okay, really, what are the underlying evolutionary mechanisms behind recruitment of additional proteins? So we began with a fairly straightforward speculation that the selective benefit is different swimming abilities. So this is swimming of 3 different species in aqueous medium. And what's really fascinating is that if we increase the viscosity by adding Ficol, Salmonella and Vibrio are completely immobilized, while Campylobacter is actually still capable of swimming reasonably well. And so we speculated that maybe this is just down to the flagellomotor. What's really interesting is that Campylobacter has fewer flagellomotors than Salmonella or Vibrio, but it's still able to swim at high viscosity. And so we started doing subtomogram averaging. This is the Salmonella motor. This C-ring, this is the rotor component we can measure, it's 20 nanometers across. But we couldn't see the stator complexes that drive rotation. Not that surprising, actually, in retrospect, because various groups, in particular groups of Richard Berry and Judy Armitage, Oxford, have shown that stator complexes in Salmonella and E. coli are dynamic and only incorporate as a function of load on the motor, and the load on the motor under low viscosity conditions required for plunge freezing is fairly low, so we don't expect to see many. This is a subtomogram average of around 300 motors, which takes a really long time to collect, actually, I can assure you. We usually get less than 1 motor per tomogram. So we expect if each of those 300 motors only has 1 or 2 stator complexes engaged, they'll just wash out in the average. Moving on, we collaborated with Ned Ruby to image the Vibrio fischeri flagellomotor. And what we saw was that the C-ring is slightly wider. And curiously, we could actually see the stator complexes here, so 13 in this case. Now, Salmonella is thought to have around 11, so it's curious that Vibrio's got a slightly wider C-ring and 2 more stator complexes. To confirm that those were the stator complexes, Ned deleted one of the stator complex proteins, and we saw that, yes, indeed, this structure that we think of as stator complexes does indeed disappear. We then teamed up with my long-term Campylobacter collaborator and buddy, Dave Hendrickson, who's in Dallas. And this is the structure of the wild-type Campylobacter flagellomotor. Much wider C-ring, and curiously, 17-fold symmetry where we would expect to see the stator complexes. So again, we got Dave to delete the motor protein complexes, the stator complexes that drive rotation. And when Dave did that, what we saw was that there's a more subtle difference, but yes, indeed, one of the major components of this 17-fold symmetry does disappear. So fully consistent with Campylobacter having a much wider C-ring and more stator complexes. So this is beginning to put the pieces together, and it's very much consistent with us thinking that yes, indeed, Campylobacter has a much more powerful flagellomotor. So moving on though, that didn't really answer what these additional components were. So we teamed up with Dave and Ned and spent quite a long time. And I mean, this next slide summarizes over a year's worth of work while I was still in the lab. And what we did is image lots of deletion mutants, just showing that you can start putting together a hierarchical order of assembly of various different proteins. I'm not going to talk about what each of those proteins are. We've got some quite exciting projects in lab analyzing these in a bit more depth at the moment. Bottom line though is we can start putting the pieces together and we can figure out which protein goes where from this combination of cryotomography, sub-Saharan averaging, and bacterial genetics. What's curious is that MOTB, which is this stator complex component, is only present in our sub-Saharan averages in the presence of the entire complement of all of these additional proteins. So what we conclude from that is that these additional proteins are requisite scaffolds to scaffold these wider rings of additional stator complexes. So building on top of that, what we're now doing is sort of collecting a zoo of different sub-Saharan averages of lots of different motors and contextualizing that against the phylogenetic tree. So, and you can see that you've got this progressive build-up as you go deeper into the tree of wider and wider rings of additional stator complexes and wider corresponding C rings, right? So I'm not going to go into that. There's a lot of very interesting evolutionary biology there that's much more complicated than I would like it to be, but c'est la vie. We're all familiar with science being more complex than we thought it would be. Now, putting it all together, what we realize is that we've got all of these structural components and some very basic physics here. You can calculate torque, right? Torque is the product of the lever length, which is the radius of the C-ring, cross product with the force exerted. Now, if we make the naive assumption that the force exerted is going to be constant across species, I think that's a roughly valid assumption given that proton motive forces have shown to be roughly equivalent across multiple species. There's one additional component to this equation. That's all multiplied by the number of force supplying units, i.e. the number of stator complexes, right? So we've got that data. We've got the forces being calculated by biophysicists, or at least estimated. Our structures give the lever length and our structures give the number of force supplying units. We've got another data point here. This is not something we've confirmed the stator complexes, but we've got the C-ring radius, and we can see there's a 16-fold symmetry exactly where we would expect to see the stator complexes. So this is another valid data point. So we've got 4 data points here, and what we can do is plot the calculated maximum torque along the x-axis from our structures and plot that against biophysicists' estimates torques on the y-axis. And what we would hope to see is a linear perfect correlation. And I have to say I was shocked that the correlation was this good, but it's extremely good. So the Vibrio species, so Vibrio is driven by sodium, not protons. So I glossed over that earlier. 3 millimolar sodium is the relevant sodium concentration that gives a sodium motive force roughly equivalent to a proton motive force. So this is beautiful, right? Sub-Saharan averaging is able to provide us with a structural explanation for how different flagellomotors can produce different torques. So we're pretty excited about that. Okay, so I haven't talked much about the methodology that we use, and I'm happy to take questions after the talk, but we've got this high throughput tomography pipeline because we have very few particles per tomogram, and what that's enabled us to do is image lots and lots of species, lots and lots of mutants, at, yes, very low resolutions, but sufficient to answer our biological question, was that what's happened across flagella motors, across different species, is that bolting on extra proteins has produced a scaffold for a wider motor, and that wider motor produces higher torque. So that's my first story. My second story, I'm going to be talking about really how cryotomography is enabling insights into flexible, transient cytoplasmic structures. And this is really something that I think is completely inaccessible to traditional structural biology techniques that involve purifying proteins. And what we're going to be focusing on is these cytoplasmic components in the lower half of this diagram. So I showed you earlier, deletion of fly eye led to loss of fly eye. Right? So this was work from my postdoc. Second paper from my postdoc was, I was a contributor to a big paper from the lab of Susan Lee, who's at Oxford. Many of you will probably know Susan. And so Susan saw that we had shown that this was fly eye and approached us to test a speculation that they had that this density may be FLHA. Now we had pondered whether this structure, this toroidal structure between fly eye and the flagella motor is FLHA or FLHB's cytoplasmic domains. These are two proteins that have transmembrane domains and cytoplasmic domains. And Susan had determined the crystal structure of a homolog of FLHA. I won't go into where that comes from, but it was we're pretty much dead certain that it's going to work in exactly the same way, was a 17 nanometer diameter torus. Beautifully, we've got a 17 nanometer diameter torus in our tomogram. And so what we did in collaboration with Susan was image this, and we truncated this cytoplasmic, soluble cytoplasmic domain of FLHA and re-imaged the motors. Now, the beauty of subtomogram averaging is that we can be capturing potentially transient, fragile assembly intermediates that we might never be able to purify. And what we saw was that, yes, we delete this cytoplasmic domain. Indeed, that disappears, but we can still image structures inside the inner membrane. None of those extracellular components assemble, not surprisingly, because FLHA is absolutely integral to type 3 secretion system function. And as a control, our other speculation before Susan approached us was that it was FLHB. We imaged FLHB, cytoplasmic domain truncation, and that toroidal structure remains. So that's beautiful. A central thing that this threw up that some of you may have realized is, well, how is fly eye suspended there, right? We delete this density, fly eye disappears. You delete FLHAC, fly eye remains there, apparently suspended in mid-cytoplasm 20 nanometers from anything else. And so if you dig a bit deeper into some of the background into this, fly eye is homologous to a part of the F-ATPase, specifically beta subunit. And there are two other components of the flagella motor which are also homologous parts of the F-ATPase. Fly H was something that caught our attention because this is a homolog of the B and delta subunits as a fusion. Now the B and the delta subunits form the peripheral stator of the F-ATPase. And so there's some precedent to think that fly H performs a similar role in the flagella motor, sort of anchoring fly eye to something. Now, I've shown you this image before. We've got different radii C rings in different species. And what's really interesting, and this was the work of Steve Johnson from Susan Lee's lab, was that you can plot C ring diameter against the sequence length of fly H. And when you do that, what you see is again, a beautiful correlation. The fly H length increases with wider C rings. So that provokes the hypothesis that fly I is held by a fly H linker up to the C ring, and that fly H linker has to be longer for wider C rings. So the work of Tague Matthews-Palmer and Louis Henderson, who are both PhD students in my lab, built on some of this. This is the wild type flagella motor. What we did is we deleted fly H, and when you delete fly H, which we speculate links fly I to the C ring, indeed, you lose fly I as a density. Fly I is still present as a protein in this species, in this mutant, but it's not able to integrate without FlyH as a linker. So this was a part of the conclusion from the paper that we contributed to from Susan's lab, was you can start building this molecular model and start speculating actually about the molecular mechanisms of type 3 secretion, that FlyI is bonded by FlyH up to FlyN, which is a C-ring component. And what FlyI is doing, which is an ATPase, homologous to the F-ATPase, is that it's taking export substrates and using its ATP hydrolysis activity to then pump them into an export chamber between the cytoplasmic and transmembrane regions of FLHA, where they are then exported by FLHA's proton channel activity. So since then, we've been doing further work on this, and this was published earlier this year. This is the wild-type Campylobacter structure. Again, this is the work of Louis and Tague. So in the wild-type structure, what we think is that FlyI is anchored by these spokes. FlyI is the sphere in the middle of this cartoon. And you can delete parts of the C-rings. This is deletion of FlyN. What's interesting is that FlyN is probably not binding to FlyH. And so FlyI can still incorporate because it's binding to another protein called FlyY and FlyM, but it's destabilized. And so if you look at the density of FlyI in this subatomic average, it's much less distinct. It's a small blob. And we think that's because when you delete FlyN, the whole thing becomes wobbly. And so FlyI is rocking back and forth instead of being stably anchored because the C-ring has been destabilized. And so when you see that, when you average hundreds of motors, you get this much smaller density where you only resolve FlyI right in the middle of the symmetry axis. And then if you delete FlyM, which is required for FlyH incorporation, what you see is you see no FlyI. And so what we think is when FlyM is deleted, that then impacts the rest of the C-ring, and so FlyI cannot incorporate at all. So you can start then building speculative molecular models about how this whole structure puts together. And I really think that this is something that's only cryotomography can contribute. A final story that's not published yet, but we anticipate, or we hope will be accepted any day, is work from a postdoc in the lab, Florian Rossmann. And so this is the Salmonella motor. Florian started getting interested in Caulobacter crescentus, which is another bacterium. And so he was very interested in transient binding of proteins that control the flagella motor. And so he imaged the Caulobacter crescentus flagella motor. That was nice. It added to our portrait gallery of different flagella motors from our zoo. This is an isosurface rendering of the Caulobacter motor. It's got this very interesting cogwheel structure up against the outer membrane that we have no idea what it is, what it's doing. So all sorts of exciting questions there. And he's then overexpressed a signaling protein that binds to the C-ring. The signaling protein is called CLE-D. And in isosurface, to gloss over many of the nitty-gritty of the results, what he sees is that CLE-D clearly saturates the outer ring of the C-ring. So again, this is transient. We've had to overexpress CLE-D, various other little genetic tweaks to Caulobacter in collaboration with the lab of Urs Yanal in Basel. But really nice, we can actually see transient binding of a protein within the cytoplasm to the flagella motor. So just to summarize that section of the talk, tomography has enabled us to image all of these transient and dynamic processes, deciphering fragile in situ structures, sort of working out through a process of elimination and logic where FlyH must be, imaging destabilized mutant structures that there's no way we would be able to purify, and imaging transient binding processes. And a pretty straightforward conclusion here, and I think I'm probably preaching to the converted here, but being in situ, what tomography is enabling us to address just previously inaccessible biological questions. And so that really is where my lab is aiming, is to really push at getting inaccessible biological questions. I did have a couple of slides on future directions. I think in terms of time, I'm not going to go over them beyond saying that I've sort of maybe become even renowned for saying that I don't care about resolution. I care about biology. I'm not that interested in getting very high resolution. But that said, most of our biology has got to the point where we've answered the questions we can at the current resolutions. So the next step is to push resolution. So we've got some exciting projects bubbling up in the lab to massively increase our resolution. We'd like to be seeing sort of hopefully around nanometer resolutions in the coming years of some of these things, and that's going to be able to start asking really specific questions about structures, and we hope to be getting at questions about, okay, well, how did these biological systems evolve? What happened to evolve new protein-protein binding interfaces when additional proteins were incorporated? So just like to say thanks to various people from my lab. Bonnie, Eli have been postdocs on various projects. Josie was a PhD student who's moved on to a postdoc with Kai Grunwald. Taig and Louis contributed to some really interesting subatomic averaging results to this, the C-ring structure project. The fluorescent microscopy I showed at the beginning was the lab of Taka, Yuki Nishizaka in Japan, Campylobacter and Vibrio genetics from Dave Hendrickson and Ned Ruby, and various other friends, buddies, collaborators, last but not least, including Peter Rosenthal at the Crick. I've got a satellite group at the Crick in Peter's lab, Ariana Brigel in the Netherlands, Kai Thorman, Kelly Hughes, and Bertus Beaumont. Various sources of funding, and I hope I've got time for a couple of questions. Many thanks for listening, everyone.
Okay, thanks very much, Morgan. Yeah, I think we're running a little behind. We've got time for a few questions, I think. So the first one I'm going to pick is, do you have any idea, so this is from Natalie Al-Hatobi, do you have any idea how the flagellar proteins, such as the flagellins, are exported through the type 3 secretion system?
Hi, Natalie. Great question. That's the holy grail of type 3 secretion. So I think this work together with Susan suggests that fly eyes are unfolding the flagellins that are then being maybe held in a cavity between the cytoplasmic and transmembrane regions of FLHA. Then it's FLHA's transmembrane region, which has a probably is a proton channel, which can somehow then start exporting stuff. But we need a structure of FLHA. That's a holy grail. Many people are working on that. We don't have it yet. So watch this space. It'll happen soon, I'm sure.
Good, good. That's promising. So a more specific question, I think, from Zhu Shiwei. Why is there no outer membrane density in the case of delta FLHA?
Hi, Shiwei. Great question. So that's because the Campylobacter flagellomotor staples the outer membrane via its P-ring and via its basal disc. And so if you abolish those, the Campylobacter outer membrane is just completely floppy. And so if you image lots of motors and average them, you don't resolve it at all. Another completely different interest in my lab is periplasm structure. And it turns out that the poles of many bacteria have no regulation of the width of the periplasm. And so without the flagellomotor, the periplasm is just massively varying in width.
Okay, good. That's very clear. One from Jolene Dixon. Do you know if non-motile flagella have all the motor components? For example, the hair cells in the inner ear?
Hi, Janine. Fantastic question. Flagella and cilia are completely unrelated. Eukaryotic structures, I refer to all of them as cilia for clarity when I talk. Totally different, so it's totally unrelated to the bacterial flagellum. I've got a a review, an epic review that's many tens of pages long, just published in FEMS Microbiology Reviews, in collaboration with other people, that compares flagella, eukaryotic cilia, sometimes called flagella, and archella, which are the equivalent structure in archaea. It compares and contrasts the three of them and how they evolved. So recommend you check that out if you were more interested.
And I think a final question. There are several questions about high throughput tomography, and I think some of them require a whole new lecture. But can you briefly say, how do you make all of this high throughputs? Because you seem to have lots and lots of different systems.
Yeah, so as some of you are aware, we've got a pretty interesting system at Imperial. We don't have a Krios. Is my screen still? You can still see my screen?
Yes. Yeah.
So this is our setup at Imperial. It's an F20 with a robot that can refill it. So we this automatically refills through a funnel the sample holder, and so we can run overnight like that. We run Leginon. I'm eternally indebted to Bridget. I think Leginon is incredible as software. So even on an F20, we can collect a tilt series in 15 minutes. So yes, I know that will improve in the future with a super stable stage. We don't have a super stable stage yet, unless you're reviewing our application for Talos Arctica, in which case, please encourage it. So we use this, and we basically have continuous runs of 48 hours or so. And so in 48 hours, we can collect 200 tilt series. On a good day, that means 200 particles. And we just use weekends when we have big projects to do, and obviously many years of work. So Leginon is the key. And then we have an automated reconstruction pipeline, which is deeply, deeply hacked, based around iMod, Tomo3D from Jose Fernandez, and Python glue scripts.
Okay, thank you. Right, I think we should move on. So thanks very much for that.
Thanks, everyone.
Good unshare.